Hello and welcome to our podcast Pager. I'm George Milner.
And I'm Stan Dale.
Tune in to hear us talk with some amazing people about the subjects changing medicine today.
From stem cells to mapping the brain and medical ethics, we hope to give you a glimpse of the bright future ahead through discussions with some of the people making it happen. Hello and welcome to the Pager interview with David Sinclair. This episode, we're going to be talking about the processes which underlie why we age and how we might be on the cusp of being able to reverse that process. David is really the perfect person to speak to about this topic. His lab is dedicated to understanding aging, and a few months ago he published a best-selling book on essentially what they've discovered. The book's called Lifespan. The fact that aging might be reversible is quite remarkable, and the reality is we're probably all going to be living a lot longer than any of us really anticipate. Just a quick note from us on the podcast. You can expect episodes to be released on a weekly basis each Sunday, but just make sure you subscribe on whatever platform you're listening on so you don't miss out. Without further ado, I give you David Sinclair. 3, 2, 1, and we're live. Today I'm joined by David Sinclair on the Pager podcast. Thank you so much for joining us.
Hey, thanks, Daniel.
Now, you recently published a book called Lifespan, which is about sort of your own information theory of aging. So, to kick us off, would you mind talking to me a bit about what you describe as the information theory of aging?
Yeah, sure. So for most of the 20th century, scientists were focused on DNA damage as a cause of aging, and that led to this craze, as I call it, around free radicals and trying to prevent damage to the genome. What I have realized over the last 25 years since I was in my mid-20s studying yeast cells is that more than likely, it's not that we lose genetic information. We lose another type of information, the epigenetic information. And what's different about those two types of information in the cell is that the DNA is digital, whereas the epigenetic, the systems that control how to read the genetic information, is analog. And that's key because analog information, as anyone who's probably over the age of 20 will remember, is terrible at preserving information. And I think the loss of that analog information over time leads to the loss of cells' identity, the loss of their function, senescence of cells, they turn into zombie-like little creatures, and ultimately that's what leads to aging itself and all the diseases that come along with it: cancer, heart disease, diabetes, and dementia.
So I suppose whereas all of the cells in the body have all of the genetic information to make any protein, what's important about the epigenome is like controlling which proteins are actually made in each cell type, which gives them that sort of characteristic, cell type.
So that's exactly right. The epigenome was first studied in earnest in mammals during embryonic development. And a fellow by the name of Conrad Waddington in the 1950s proposed what he has now termed, or we've termed, the Waddington landscape. And it's a metaphor for how a single fertilized cell becomes a collection of 26 billion cells in a young child. And we take it for granted, but it's the epigenome that directs those cells to become what they are. But what Waddington didn't do was to extend life beyond birth. What is it that happens to the Waddington landscape as we go through our 20s, 30s, into our 80s? And my theory says that it's that landscape that holds the cells in place and stops them from jumping across from one valley to another. That's what goes awry. The cells jump and move across from one valley to another, and that's what causes them to lose their identity, because each valley of that landscape defines what type of cell that cell should be for the rest of our life.
So really, according to your theory, all the other sort of hallmarks of aging, the degradation of telomeres, DNA damage, unfolding of proteins, you think that can all be traced back to these epigenetic, the loss of the characteristic epigenetic profile of specific cell types, basically?
There's two answers to that. One is that the hallmarks of aging, and there are 8 or 9 of those, and they range from loss of stem cells, breakdown in nutrient sensing, protein misfolding, and the digestion of those misfolded proteins called autophagy. One is that I think this is the best explanation for an upstream cause of all of those things. linking them all together. And so that's one explanation. The other is that we have a couple of papers under review that show that if we disturb the epigenome and disrupt that information and then therefore make the cells lose their identity, we get a mouse that is 50% older based on molecular, histological, physiological measures, and even measuring the actual age, biological age of the animal, which we can do now, what's called the Horvath clock. And so that is very good evidence that this is an upstream cause of all of those downstream hallmarks of aging that people have been studying, including me, for the last 30 years. Now, we don't know everything, of course. You can't solve something as complex as aging in one book. But I do think that it explains the last 200 years of observations in a very simple and unified way. It also ties into cellular senescence because, as I mentioned, as cells become more deranged, they eventually end up being senescent. We see that in our lab. And even
Senescent cell is a cell which is sort of no longer dividing and is releasing lots of pro-inflammatory stuff, which is like damaging the surrounding tissue. Is that right?
that's exactly right. And so that's why they're called zombie cells. That they check out of the system, they stop dividing, and they wreak havoc in the body and make other cells age prematurely too. And the thought is that if we can delete those cells from the body, we could be somewhat rejuvenated. But what I'm saying is that I think we understand why that occurs in the first place, and even how to prevent it.
I heard you speak to Peter Attia on his podcast, The Drive, about like Horvath's clock. Like you're saying, this is essentially you can do with this clock is look at the epigenetic profile of a cell, specifically DNA methylation at like some very specific sites in the genome, and from that, like work out with a remarkable degree of accuracy how old these cells are.
Yeah, it's been a huge breakthrough for the field because we didn't really know how to prove an organism was actually old or a cell was actually old. We could measure telomeres, but often cells become old and senescent even without losing their telomeres. So it was far from perfect. So this new clock is seemingly unbiased and it measures the rate of aging, not just in humans with a blood test, but in mice and other animals such as bats and even sheep. So it's a remarkable discovery. But the question really is, what's driving that clock? And that's what my theory explains, that the unraveling of how the DNA is packaged over time leads to changes in how the DNA has chemical marks on it. You mentioned DNA methylation. And those DNA methylation marks are a very core, very deep level of what we call the epigenome that regulates how the cell reads the genes. And the slow loss of that original embryonic program from when we're young, I think, is what's driving aging.
What is the actual assay underlying this clock? Like how are you actually looking at methylation at different points?
Yeah, it's really simple actually. We just protect those regions or digest those particular regions. We modify them chemically. And depending on how they read in the DNA sequencing machine, we can tell whether a site has been altered by the methylation treatment. And so what we do is we get a different letter in the genome where the methylation used to be. And by measuring thousands and thousands of changes in the methylation pattern across the genome, we plug those numbers into, like you say, an AI algorithm. We do machine learning, and over a matter of days, it spits out the best combination of a few hundred of these sites that can, with great accuracy, predict the age of the sample. And we actually split the sample up into two. We train the algorithm with half the sample, and then the rest of it we test using our artificial intelligence derived algorithms. And often they are 95 or more percent accurate to be able to predict the age of the tissue or the animal. And
I
that's
think you
what we
mentioned
call.
in a previous podcast that you can even use this to sort of predict how much somebody's smoked to like a very high degree of accuracy in their life.
Well, yeah, Steve Horvath is publishing every week, it seems, on a different cohort of people or animals and just coming up with these remarkable discoveries. Smoking, being overweight, these are all bad things. And that's not news. I mean, it's not new either, but it tells you that this clock is extremely accurate. And he can also predict the with pretty good accuracy when someone will die within a matter of years. And
Which is
using
quite spooky.
it's freaky when that actually when you realize that that's now possible. And I don't know about you, but I'm even hesitant to do that test because then what do you do when you know your death date?
Well, actually, that leads me really well onto the next thing I wanted to talk about, which is using a clock like this. Perhaps we could then more quantitatively look at things which might potentially slow aging or extend lifespan, supplementing NAD and metformin and also resveratrol. But I think to talk about two of those things, NAD and resveratrol, we probably need to go back and talk about the role of sirtuins in controlling this epigenetic profile of cells. So would you mind telling me a bit about what sirtuins are and sort of what they do in cells?
Yeah, sure. Sirtuins are, think of them as the protectors of our body. They evolved very early on in life. They're found in plants and fungi and ourselves. And they respond to damage and they help fix it. And we found that by putting more copies of sirtuin genes into mice, they can live longer. And we think that they're also responsible in part for the benefits of a healthy diet, of fasting, and exercise. So these are a major discovery in the field. There are other genes that they talk to. There's ones called AMP kinase, there's one called mTOR. These genetic pathways form a surveillance network that sense when our environment is tough, when there's adversity coming, and they hunker down and allow us to survive. Unfortunately, we live a life of guilty pleasures, most of us, and we often don't exercise, and we eat way too much. We're never really hungry, and that means the sirtuins they become complacent. They're not always active, and that is, I believe, one of the main reasons we become susceptible to cancer and diabetes and heart disease more than we would otherwise if we were stressing the body biologically. And what just the link to the epigenomic theory, the information theory of aging that I've put forth in my book. Sirtuins, we started working on them many years ago, 1990s, mid 1990s, when I was in the lab of Lenny Guarente at MIT. We discovered that these sirtuin genes make enzymes that control the epigenome, and that's one of the main reasons that they can extend lifespan. And what's interesting about sirtuins is they're named after a gene in yeast called SIR2, SIR number 2, and what's cool is that name is silent information regulator number 2, and the key is
the information theory of aging really is a good fit for the kind of genes that we're talking about, isn't it?
well, that's exactly right. And what's funny about it is that it's been in plain sight for the last 25 years since we first published it, but only now am I putting it all together into a theory, into the information theory of aging.
It's interesting. It seems like a lot of the things which are relevant to cellular aging, you mentioned mTOR and MAPK, which I think are proteins which are involved in sort of metabolic sensing. So they detect, how much nutrients there is in a cell's environment, right, and sort of then affect the processes the cell does as a result of that. Like rapamycin, this is a drug that was originally an immunosuppressant, isn't that right, for people who had surgery, but now has sort of emerged as potentially this big player in slowing aging. It's just interesting that, like you said, a lot of these things kind of come in under the radar or have been around before, and taking a second look at them has kind of been fruitful.
Well, right. So rapamycin is interesting. It was first discovered on the island of Rapa Nui, or Easter Island as we know it, and it was put away for many years. It was originally thought to be helpful with a toenail fungus, but then because there were plenty of safer treatments for toenail fungus, it started to be used for transplant to prevent transplant rejection. But then and actually David Sabatini at MIT gets a lot of credit, and Michael Hall for his work in yeast. They discovered that TOR is a sensor of amino acids. So when you eat a lot of meat, if you eat a giant steak, TOR will be switched on and makes the body grow and basically become more fertile. But that's what you do when times are good. But what happens is that's at the expense of hunkering down and protecting the body, because you can't do both at the same time, or at least the body doesn't like to do that. And so rapamycin was tried as an anti-aging longevity molecule, and in many organisms, from yeast cells to mice, and maybe in humans, we have evidence that it is an anti-aging molecule that's on the market already, albeit with obviously some side effects that we don't want.
What are the side effects?
Well, if you take a high dose, you're going to trash your immune system. So people are under medical supervision trying small and intermittent doses of rapamycin. What's good about these other two pathways, AMP kinase, AMPK, and sirtuins, along with NAD, which we'll talk about, is that they seem to be very safe, unlike rapamycin. And so NAD boosters, we haven't seen any downsides yet in 2 years of clinical trials here at Harvard, nor in the mice that we've been feeding these molecules for over a year. Actually, probably the last 3 years in all.
So just to recap, these sirtuin genes are very important in controlling the epigenome of a cell, and also I think are responding to damage to the DNA within a cell, which actually prevents them from performing that first function. If we can improve the function of these genes, then you can actually slow the aging process. And so when we talk about things like NAD, that's relevant because it's sort of a cofactor. It's the substrate for these proteins. So it sort of helps them perform their role. Is that right? And that's sort of where this idea of
Yeah, you got it. These are, we talk about these being sirtuin genes, and there are 7 of them in our bodies. But just going back to, if we want to remember high school genetics, those genes make proteins. And so we interchangeably talk about the sirtuin genes and the sirtuin enzymes, which are proteins. Now, I think the enzymes are much more interesting because they actually do something. The genes are just the code for the protein. Yeah. So the enzymes, they're some of the most remarkable enzymes in the body. They're very complicated in their structure. They sense the levels of NAD in the cell, which indicate how much you've been eating, whether you're tired, whether you've been exercising, even what time of day it is. And they become active depending on the level of NAD. And the more NAD, the better for these sirtuins. And the enzymes, what they do is they're like little traffic cops that tell other proteins to go do a good job, go repair the telomere, go repair that DNA, go burn that fat. And when you activate them with NAD or you put in extra copies of genes into mice, they have those benefits and they don't get many of the diseases of aging until much later. And we can activate them either by raising NAD or giving them the molecule resveratrol, which we first worked on, the molecule from red wine.
how about exercise? Does exercise boost NAD levels, or does that come in a different way?
It does boost NAD levels, and we know a lot more in mice, but we know a little bit in humans that you can say a marathon runner will have about 40% higher NAD levels circulating and in their tissues. And it's also true when you're fasting. It's also true when you wake up in the
Well, I
morning.
didn't realize that.
Your
I
body
didn't realize
has
that fasting increased NAD levels. I always thought of fasting as tying in with these sort of metabolic sensors, mTOR and MAPK, but I didn't realize that fasting actually like boosts NAD levels. That's pretty interesting. It's pretty interesting.
Yeah. Well, the interesting thing is they're all talking to each other, and unfortunately, scientists like to um, promote their own work. That's their job. That's our job. And often in the media or in papers, you just hear mTOR is the only important one or AMPK is the only important one. The truth is that these genes and these pathways that we're talking about today, you can't move one and not affect the other. These are levers that are connected. And exactly. So when you exercise, your NAD will go up, and then the sirtuins will turn on AMPK, and you get a similar benefit to what you would have from taking a drug like metformin, which is an artificial AMPK activator.
So we've spoken a little bit about methods for slowing aging. Are we now at a place within the field where you can more quantitatively sort of look at the efficacy of different sort of interventions or different molecules? Because it seems quite qualitative up until now.
Well, you're exactly right, and it's accelerated our lab's research tremendously. And in my book, and you'll know this having read it, we've made some extraordinary breakthroughs that aren't even known to the public yet, or at least they have been since my book came out a week ago. But I've put some remarkable findings that use the Horvath clock to not just understand why we age, but how to reset that clock. But the most important thing about this clock, um, besides it being helpful and more accurate, is that it used to take us years to figure out if we were affecting the aging process. In a mouse, it would take us 3 years to finish an experiment, sometimes longer, because a mouse lives up to 3 years and longer if you're successful in this field. But in humans, imagine how long that experiment would be. It would take a lifetime.
Yeah.
But now we have a way, within a matter of months, of looking whether you've changed the course of biological age. And in fact, I was pretty excited by a small but intriguing study by Fahey and Horvath. Horvath, the clock guy, my friend Steve at UCLA. They published it was about 10 days ago, that they ostensibly could reverse biological age and give people another 2.5 years of life with a combination treatment of three molecules that they thought might impact aging.
And I suppose the other advantage of, like you said, being able to do these experiments over a shorter period of time is you just get less confounding factors, right? Like if you did an experiment over somebody's entire lifetime, like there are lots and lots of different things affecting when they die. So I suppose, yeah, you can control it more if, you can more quickly get this information about how the aging of cells is affected. Out of interest, what were the three compounds?
The three things were growth hormone and DHEA and metformin. And the rationale there was that growth hormone was spiking the glucose levels, which you don't want for longevity. And the theory was that those other two molecules could keep glucose levels down. Metformin is definitely known to do that.
Yeah, so why were they giving growth hormone? Because it seems like it does the opposite of kind of what you want.
Yeah, you're totally right. And so it wouldn't have been my first choice, but
hey, it works.
well, we see, 2.5 years with 9 people. It needs to be reproduced, of course, and with more people. But the idea is pretty interesting that it might be possible. Growth hormone. Let's talk about that because it's on a lot of people's minds. Many people take not many, but there's a subset of people who take growth hormone as they get older to replenish what's lost, and they do see improvements in overall energy and I think muscle growth. Testosterone is another hormone that's used to replace the loss in men, particularly, and so that's all good. I wouldn't say having more strength to prevent falls is a bad thing, but in extensive tests, there hasn't been any improvement in longevity or any evidence that until now that aging is affected.
Well, like you say, it seems like the body is either there's an equilibrium between sort of longevity and growth, and like to do both of these things at the same time is like very difficult, if not kind of impossible. So, yeah, I'd be interested to find out like what the effects of testosterone replacement therapy are on like longevity. Does it ramp up your metabolism and in doing so sort of increase the rate of aging? I don't know.
Well, yeah, they didn't do the clock because it wasn't available, but there was no evidence for long-term benefits. But yeah, you're right. Now,
it must be a very exciting time.
100%. Now, what's happening is so Steve Horvath is overwhelmed because uh, people who have freezers full of all of these blood samples going back years can now determine those ages, and there's such a demand. One of the things we're working on in my lab here at Harvard is to have a very cheap test for biological age based on the methylation clock, because right now it's at least a few $100s for each sample. Be nice if it was down to just $1 and everybody could get this test done and come back every year and have it done in their doctor's office to see how they're doing.
Now, is the technology behind these assays and this clock, are they patentable? I'm curious. Like, would he be able to patent?
The algorithms are, for sure. and the methods that we're developing will be. But we just to be clear to you and to your listeners, my lab's not about keeping things from people. It's the opposite. We make a discovery and very quickly we get it as fast as we can into people's hands. Whether it's a blood test for an infection, which is one company, a treatment for wound healing for chronic wounds that came out of the lab. So we're innovators here, and the only reason we file patents is so that investors will back us.
So something else I really wanted to talk to you about is we've spoken a bit about your kind of general theory about why people age and like potential methods of slowing aging. I suppose what's really novel in your book is this idea of potentially being able to reverse aging. And you use this analogy of, you use an information theory to explain that when information is being sent between whatever you want to call it, a sender and a receiver, there ought to be like a master copy maintained so that the received signal can be compared to like an original to check if it's degraded or not. And this can be applied to talking about the epigenome and like methylation of genes. What I really don't understand is like, what is this master copy?
Well, that's the Nobel Prize, and we're not there yet. But it's remarkable that it's possible to reset the cell's age somehow, and we think we've figured out one safe way to do it, apparently safe. And as you mentioned, it's a combination of 3 of the Yamanaka factors that were previously shown to make stem cells out of our adult cells. I could take your skin cells and turn them into any other cell type I wanted. I could make a small organ, I could make new skin, I could make immune cells. And you have to use these reprogramming Yamanaka factors. Now, we've been looking for a way to reset the epigenome for many, many years, ever since yeast, actually, last 20 years. I haven't talked much about it because it was under the radar. We've been actually working on a particular type of mouse that we can accelerate and reverse aging in for a decade, and I'm only just revealing this to readers. So definitely go read the book if you're interested in learning ahead of the scientific field.
Your work's shown that you can introduce certain factors called Yamanaka factors, which have previously been used to kind of turn cells back into a more primordial form, like back into stem cells which can differentiate into different kinds of cells. And if you introduce those factors, I think using viral vectors, essentially you can get these cells to like regain their youthful methylation profile. Sort of how do cells, I suppose what I'm trying to ask is, how do cells know what their original methylation profile was?
I was right, as you know, I was writing down what it was like to discover this just 6 months ago. And in the end, my editor said, "Stop adding this. We need to publish this book." And so it's come out really hot off the presses. You know, our heads are still spinning with the realization that there's a backup copy of youth in our cells that can be accessed. We didn't know that was true 2 years ago.
It must be an incredibly exciting thing to know, but also a very difficult thing not to share with other people because it is like so fundamental.
Well, exactly. You're right. Let's talk about the fundamental. What does this mean for humanity? What it means is that an old person still has the information to be young again in their bodies. It's just that the cells need to be told how to access that information. I make the analogy in my book about our genome being the music on a DVD or a CD. And for the very young in the audience, those are things we used to put photos on, the ancient technology from the 1990s. But it right now it's the best analogy. That DVD is the storehouse of all the information to make our bodies. Call it a symphony. But as we get older, we get scratches on that DVD. So the reader, our cells, cannot read the right tracks or songs at the right time. And it's a cacophony eventually that at the cellular level causes cells to lose their identity. But what if there's a backup of that DVD? Or even better, what if we could just polish the DVD and get the information back again and you could have that symphony again? We think that that's true. We think that's possible in our bodies and have some early evidence that we've just posted online that says that at least in the mouse, in their eye, we can reset the age of cells and get back their function so that old mice can see again like they were young.
Mm. It's occurring to me just now, actually. Your cells can essentially tell whether something is paternally or maternally inherited. Is that right? There's sort of genetic imprint. There's genomic imprinting, isn't there? That could be an interesting area to look at to better understand how cells might know when sort of certain epigenetic changes to cells happen. Sort of what might have been originally there, and then what might have happened throughout life.
Yeah. Well, you bring up two important things. One is that how your parents lived can be transferred to the offspring through epigenetics. So, as the mother, at least in rodents, if you feed a mother a high-fat diet, she gets obese, then the offspring will be prone to diabetes. And we've been using NAD boosters to try and help those kids, and even in utero to see if we can prevent that. So far, things look promising. And then the second is that because aging, I believe, is mostly epigenetic, and therefore it's not just slowable but reversible, a lot of how we live our life does determine our longevity. If you look at twin studies and large populations, the conclusion is that our longevity and our health into old age is 80% how you live your life and only 20% genetic. And that's a remarkable realization, remarkable possibility, which gives me hope that everyone can take their longevity into their own hands.
Yeah, it's really, to me at least, it's quite surprising that genetics only contributes about 20% of that. So, like you said, quite an empowering message, actually, that you can sort of take into your own hands the course of, or length, very specifically, of your own life.
But what a wonderful thought that we used to think aging was something that was quote unquote God given, inevitable, natural, therefore acceptable. And in my book, one of the main things that I make a case for, and many people who have read it have said it's changed their lives in the way they view aging and the arc of their own lives and their family, is that we should not accept aging. We don't have to accept aging as we know it right now, and that we can each of us do something about it right now.
I think that realization has the potential to change the way that humans quite fundamentally live their lives. And one thing I want to get onto later is, I suppose, the dark flip side of what we're talking about in terms of people being able to take their own aging into their own hands, which is that as these things become better understood and products become available, who are they first going to be available to? People with means. And you could see some sort of large divide, as you spoke about in your book, between sort of the haves and the have-nots in terms of our ability to extend our lifespan. But quickly before we get onto that, I wanted to talk to you about something that you and Peter Attia were talking about on his podcast episode with you, which is that it seems like with the introduction of these Yamanaka factors, these factors which are restoring this youthful profile, there's sort of a balance in that if you introduce too much, you actually end up turning these cells, winding the clock back too far, back into sort of a state of stem cells, and you can get, I think, tumors occurring, tumorigenesis. How is that something? Is that so? Sort of, is the introduction of these factors something that you're going to continue to sort of like dial down to a more specific amount, for want of a better word?
Yeah, well, that was the trick, actually. We were looking for many years on how to reverse the clock and restore the epigenome as though it was youthful. Another way to think of it was to get those Waddington balls back into the valleys that they came from so cells would remember what they should be. Or even simpler, imagine a scratch DVD. What is that polish? And we went through many different factors. We were trying factors from early embryos. And there was a remarkable paper, one that I suspect could win a Nobel Prize, for Juan Carlos Belmonte at the Salk Institute. He and his team, Dr. Ocampo was the first author, who also deserves a lot of credit for this. They had a mouse where they could turn on 4 of the main Yamanaka genes in a mouse. And this mouse, by the way, was not a normal mouse. It was a rapidly aging one that is a genetically modified mouse that ages prematurely, called the lamin A mouse. In any case, they turned on the Yamanaka factors, and first of all, they got a dead mouse. After 2 days, they all died. Okay, so that's no reason to get excited. But what they did that was clever and brave was to turn on the genes for 2 days but then switch them off again for another 5. And by doing that repeatedly through the life of the animal, they got a 40% lifespan extension. Now, there are a few problems with that study. It wasn't perfect, but it was a pretty interesting one. First of all, you can't have a therapy where you almost kill a patient. Second of all, was it working just because it was stressing out the mouse, or was it working because it was truly reversing aging? They had no idea. They were looking at tissues under the microscope. They didn't do a lot of molecular analyses, and they didn't measure the Horvath clock, surprisingly. Now we've done that. We've made a few improvements. What we've realized is that 3 of the Yamanaka factors are safe and 1 of them is not. So we've taken out the MYC gene, which is a cancer-causing oncogene. We've improved the delivery, so we don't have to genetically modify the mouse from birth. We can use, as you said, viruses to deliver it to any part of the body in a mouse, or eventually, hopefully in a couple of years, in a person. And then the other critical thing was we needed to ask the question, does age reset and do cells regain their identity? Is that how it's working? This would be the equivalent of everyone thinks that the Horvath clock is just a clock on the wall. Scientists think that it's an indicator of age, but it's certainly not the reason we age. Our experiments indicate, and I propose in the book, that the clock is actually the age, so that when you turn the hands of the clock backwards, it doesn't just indicate that you're more youthful, it actually makes you more youthful. Yeah. The clock is actual time, which is mind-blowing. That's a big shift in how we think about all of this. And we even understand some of the mechanisms behind that clock. We don't know all of it, but we do know that there are little cogs back there called TETs. And TETs are the enzymes that take off these methyl groups from the DNA, allowing us to reset the clock. Now, we don't want to do it too much. We turn on these genes very low levels, and we turn them off when we've restored eyesight or regrown the optic nerve, because we're worried that if we keep them on for too long, there might be some negative effects. But that said, we've had some mice with these 3 genes on for over a year now, and there doesn't seem to be any evidence of cancer. So we're cautiously optimistic that we've made a breakthrough here.
Yeah, and I think it's worth pointing out to listeners that we're talking when you add together. Sorry, I can hear myself through your headphones. It's slightly freaking me out. Let me start that again. I think it's worth pointing out that, like you said, when you use the figure of 40% in mice, when you compound the effects of this gene therapy involving the Yamanaka factors, potentially with lifestyle interventions or supplements, you're talking about extending life for its decade, isn't it? You speak in the book about how you think 120, 150, these are ages which like really aren't out of reach, and also that we're going to be in a place where people are going to have their quality of life maintained for much longer. So it really is it's going to be a fundamental change in the way that humans view themselves, I think. And I wonder, like, you speak in the book a bit about what you think the implications are for the broader society, and sort of what might be the pitfalls and what might be the benefits, obviously, of extending lifespan. Specifically, I'm curious, do you think that people are going to become more risk averse if we realize we essentially might not age? So, I phrased that awfully, but I hope you sort of get what I'm going after.
I sure do. When you realize that our generation could live a decade longer than we thought we would, by doing things now that I talk about in my book, and things that are just coming online, and then eventually reprogramming the body to go back a couple of decades, that I believe is within the realm of possibility within our lifetimes, then we're talking about a very different life than what previous generations had access to. Now, what does that do? Well, it gives you a different perspective on retirement, on savings, on the potential for having a new career, and even what you can do in your retirement, because we're not just going to be living longer, we're going to be younger for longer. We're going to be able to travel the world. We're going to be able to hang out with our grandkids and even our great-grandkids. But it also means if we one day, and I don't know if we will ever get to 150, but I think there's no reason future generations couldn't do that. What does that do about your fear factor? What about, is it safe to cross the street if you live that long? Now, I don't think that that's going to change quickly. I think if we lived a few thousand years, yeah, that would change the math. But I think overall our lives would be pretty similar in the way that we've changed over the last 100 years, going from an average of about 50 years of life expectancy from birth to now, where most of us expect to reach 80. We are more cautious for sure than we used to be. We wear seatbelts and wear helmets. And you're right that there may be some more adjustments like that, but I hope not too much, because we just don't want to take the fun out of life along with all of these precautions.
Yes, you don't want to stop living just because you're living longer. And I think you're right. I think a lot of our assessment of risk is probably at quite a subconscious level. So it will take a long time for our attitudes to, crossing the street to change. I wonder, are there people in government, people in policy, in global health, who are getting ready for this shift in how long humans are going to be living? How is this going to change our models for how the economy ought to work? What are the social effects going to be?
This is why I had to write the book, because the world isn't ready for what's coming. I mean, I was, of course, excited to let the planet know, everybody who's listening, I hope you're excited about what you're hearing, because it's going to fundamentally change our families, but also the global economy and politics. And we aren't ready at all. Now, there are going to be some good things, as I mentioned. There are going to be some bad things. What are we going to do with a bump in population? What are we going to do with people working into their 90s and beyond?
Yes, what about?
And I have solutions.
In the book, you posit this idea of the dictator who's able to reign for 100 years and
and
right
wing or
yes, exactly,
left
social changes.
Extreme politicians at both ends will live longer. We're already seeing that. So I am talking to Congress people, men and women. I'm speaking to leaders of industry. I'm speaking also to members of the FDA here in the US. So these are regulators. Also in the UK and Australia and Israel, those are the main hotspots for this interest and preparation for what's to come. It's very early days, and most countries have no idea what's coming and what's going to be possible.
And I also wonder how popular it will be, if politicians start saying, right, we need to push back the age at which people can retire and get access to sort of social benefits. So I'm sure there'll be, even if we can see these things coming, there's going to be a whole bunch of other problems stopping us from making effective changes to prepare for it.
Yeah, right. But we will adapt, because it won't happen overnight. But one of the most important take-home messages from today and in my book is that the amount of money that you save by not having people spend the last 5 to 10 years of their lives in a sick state or in a nursing home. Instead, these are active people in the community taking care of their kids, grandkids, and even starting a new career like my father did at age 77. That's in the trillions of dollars per year across the globe. And that's money that can be put into social security and retirement, can be put into addressing global warming and saving species. There's nothing else that will save that much money except reducing the healthcare burden across the world, which is only going to increase as the number of older people increases as we go forward.
David, I feel like we could talk about the implications of this, the changes in the way we age for like another hour or two, but unfortunately, I think we are right about out of time, and I don't want to eat into any more of your day. So thank you so much for coming on the podcast. It's been a real pleasure.
Hey, Stan, thanks for having me on. I really enjoyed it.
Thank you for listening to the Pager podcast. If you've enjoyed it, then head to Spotify or iTunes to hear more.
As ever, we'd love to hear what you think, and you can reach us via our Facebook page or by email. See you next time.